Recombinant DNA technology. Recombinant DNA technology involves using enzymes and various laboratory techniques to manipulate and isolate DNA segment of interest. So when we said recombinant DNA technology is a technology that uses enzymes and various laboratory techniques like PCR, that is polymerase chain reaction, and sequencing method to manipulate and isolate DNA segment of interest. Like for example, you can use the recombinant DNA technology to manipulate and isolate the DNA for insulin production, oxytocin production, and of course, with the knowledge of recombinant DNA technology, a lot of drugs, vaccine are being actually produced, crop improvement, drug resistance, as well as the crop resistance have been developed with the knowledge of recombinant DNA technology. So therefore, recombinant DNA technology is a tool of biotechnology that employed in development of various products. These methods can be used to combine or splice DNA from different species or to create genes with new function. The resulting copies are often referred to as recombinant DNA technology. So generally, this method that are used or this recombinant DNA technology is used to combine or splice DNA from different species of organism, combine them to create a new genes with new function. And this will actually result in the formation of a copies that are called recombinant DNA technology. So generally in recombinant DNA technology, you can get genes from two different organisms. You can combine them so that to have a new functions. And that new functions usually are of health and society development. So generally the knowledge or the concept of recombinant DNA technology is employed in a lot of the generation of products that are of benefit of humans, like development of drugs, development of vaccine, crop improvement, production of genetically modified organisms, in vitro cloning, and so many other products are generated using the concept of recombinant DNA technology. And of course, look at the figure there. The genes first identified, combined with a vector, then you have the recombinant DNA technology, and then it's now transferred into a host like bacteria. And of course, the recombinant DNA technology will now amplify by the actually the cellular division of the microorganisms, which is the bacteria. So because the bacteria contain the recombinant DNA technology, and generally bacteria have a low generation time, so as they are generating, dividing themselves, the recombinant DNA technology is also multiplying. So we have steps of recombinant DNA technology, which first include isolation of genetics material, cutting of DNA at specific location, joining of the DNA fragment by ligation and uh, homopolymer tailing, and then insertion of DNA into the host cells, and then selection and the screening of transformed cells. So generally, in the steps of recombinant DNA technology first involve the isolation of the genetic material of interest. Let's say you want to isolate the DNA or the segment of DNA for insulin production. So first the DNA will be isolated, and then after isolating, then you now after isolating the DNA, then you now cut the DNA at a specific locations using restriction enzymes. And then after you do that, then you will now join your DNA of interest after cutting with a vector, and the vector is also cut with the same restriction enzymes that is used in cutting your DNA of or DNA segment of interest. The restriction enzymes are also called molecular scissors because they are the enzymes that are used in cutting the DNA at a specific fragment of DNA. And then another enzymes called ligase will also be used to ligate the vector and the DNA of interest. And after doing that, then the combined DNA, that is that of the vector and the DNA of interest, will be transformed into the cells. And of course, look at the figure here. First, we have the DNA donor, and we have the restriction enzymes which actually cut them into different fragments, and then the same restriction enzymes is used to cut the vector. So after cutting them, then you have your DNA fragment of interest, and then you have your vector. Then the next thing is to ligate them, to join them. So after joining them, you see that you have what you call a recombinant DNA molecules. And these recombinant DNA molecules will now be transformed into a bacterial cells or a yeast cells where they multiply themselves. So after you actually transform the bacterial cells with the recombinant DNA technology, then the next thing is the selection of the transformant, because you may have a millions of microorganisms or a millions of the bacteria or yeast cells, and it's not all of them that will take off the recombinant DNA molecules. So you have to select and identify those bacterial cells that actually transformed with their recombinant DNA molecules using, in many cases, we use antibiotics, because most of the transformant, they are resistance to antibiotics due to the presence of the recombinant DNA in them, because the recombinant DNA contain resistant or antibiotic resistant gene, and that is what actually make them to survive by adding antibiotics in the culture system. So after doing that, then the next thing, your transformant will be multiplying, so that at the same time your recombinant DNA molecules will also be multiplying. So the next thing is depend on what the downstream applications that you want to use the DNA molecules for. So now in the next video we are going to look